Bismillahirrahmanirrahim. Assalamu alaikum dear students. In the previous lecture we have discussed about development of embryo, and we have also discussed the second change, that is development of endosperm. Today we will take up development of seed and fruit. This is the third and fourth post fertilization change, that is development of seed and fruit in angiosperms. As we know, seed is a ripened ovule. Seed is a ripened ovule, and after ripening, seed gets converted. Ovule produces a seed, while as fruit is a ripened ovary. And the ovary after ripening produces a fruit. And in short, we can say seed is a ripened ovule and fruit is a ripened ovary. And we also know, we have seen fertilization. In angiosperms, fertilization provides a stimulus. Fertilization provides a stimulus for the formation of zygote. Fertilization provides stimulus for the formation of zygote. And in angiosperms, we have double fertilization. This double fertilization in angiosperms, double fertilization produces two structures. Double fertilization produces two structures. In angiosperms, double fertilization produces two structures: a diploid zygote and a triploid primary endosperm nucleus. And in angiosperms, double fertilization produces two structures: a diploid zygote and a primary endosperm nucleus. The diploid zygote produces an embryo, and a zygote forms the embryo. While as primary endosperm nucleus produces a nutritive tissue called endosperm. We have discussed these two, and the development of embryo and the development of endosperm. And a zygote, it produces embryo, and primary endosperm nucleus produce a nutritive tissue that is called endosperm. This endosperm provides nutrition to the growing embryo, and endosperm provides nutrition, that is food, to the growing embryo. With the growth of the embryo, the central part of endosperm is consumed. Because embryo is getting its food from endosperm, while during its growth, with the growth of the embryo, the central part of endosperm is consumed. We have seen during the development of embryo, the suspensor, the history of the suspensor, it enters into the endosperm to get food from the endosperm for the development of the embryo. With the growth of the embryo, the central part of the endosperm is consumed. And endosperm, the other part of the endosperm, the remaining part of the endosperm, it corrodes over the nucellus. The remaining part of the endosperm, it corrodes over the nucellus, which is the tissue which is surrounding the embryo sac. Endosperm, the remaining part of the endosperm, it corrodes over the nucellus. And as the embryo development reaches maturity, its further growth its further growth gets suspended or is suspended, and when embryo development is completed, when it matures, its further growth is suspended due to the development of inhibitors, and some inhibitors are there produced. We do not allow the further development of the further growth of the embryo. And another thing, abscission of funiculus, that is the stalk of this ovule, it gets cut, it gets separated from the ovary wall, that is abscission of the funiculus, and change in integuments. And when embryo development reaches maturity, its further growth does not take place due to three factors. Number first, due to the development of some inhibitors, we do not allow the further growth of the embryo. Number second, abscission of the funicle, and in the funicle, where from it was getting the food from the ovary, it gets cut, it gets separated, it gets abscised from the body of the ovule. And number third, that is change in the integuments. Now, the cells of the integuments enlarge. Lose their protoplasm, that is they become dead. They enlarge, lose their protoplasm, and develop thick and impermeable walls. They develop thick and impermeable walls. And in the cells of the integuments, they enlarge, lose their protoplasm, develop thick and impermeable walls. With this, we have seen generally we have two integuments in case of an ovule. The outer integument becomes hard. And leathery. And we have seen integuments undergo change. The outer integument becomes hard and leathery and forms outer seed coat called testa. The outer integument becomes hard and leathery and forms the outer seed coat called testa. The inner integument, if persists, that means it is not present in all, and if it is present, if it persists, forms the inner seed coat, called tegmen. The outer integument becomes hard and leathery and forms the outer seed coat called testa. The inner integument, if persists, forms the inner seed coat, that is called tegmen. With this development, with these changes, abscission, this development of inhibitors, abscission of funiculus, and the various changes in the integuments, with this, ovule is transformed into seed. Ovule gets transformed into a seed. And with this, ovule gets developed, it gets transformed into a seed. That means with this, development of seed takes place. Now, this development of the seed, it is taking place within the ovary. Wall. Ovary is found in the this ovule is found in the ovary. When ovule gets converted into a seed, the tissues of the ovary wall also get stimulated. When ovary wall sees that the ovule is getting transformed into a seed, they get stimulated. They get stimulated to grow with the development of seed. To grow with the development of seed. And they get stimulated and they also develop with the development of seed. And with this, the ovary wall produces a fruit wall. It produces a fruit wall. And outside the seed, it produces a fruit wall or pericarp. And when ovule has converted into seed, this ovary wall also gets stimulated and produces a fruit wall which is also called as pericarp. But generally we see the growth of the fruit is generally very fast. The growth of the fruit ovary, it is very fast. It is very fast. It is very fast and it grows with the growth of the seed and completely surrounds over the seed coat, and produces a fruit. In some plants, thalamus and other floral parts, these fruits which are produced from the ovary, they are called true fruits. The fruits which are produced from the ovary, they are called true fruits. And in some cases, other floral parts like thalamus and other floral parts, they also proliferate along with the pericarp, along with the ovary wall, and such fruits are called false fruits. And the fruits which are produced from any part of the ovary, any part of the flower other than ovary, they are called false fruits. For example, we see apple. Apple is a false fruit which is produced from thalamus. Apple is a false fruit that is produced from thalamus. During the development of this fruit, two types of stimuli are involved in the formation of fruit. Two types of stimuli. First stimuli comes from pollination, and second stimuli is it is received from developing seeds. And the ovary is getting stimulated two times to produce the fruit. First stimulus it gets from pollination. When pollination takes place, ovary wall is getting stimulated that it has to produce something which is fruit. And second thing, when ovule gets converted into seeds, and when seeds are developing, the second stimuli is received by the ovary wall to produce the fruit. With this, the seed and fruit gets developed. Let me repeat. Development of seed and fruit is our today's topic. Seed is the ripened ovule, while as fruit is the ripened ovary. We know fertilization provides stimulus for the formation of zygote. And in angiosperms, double fertilization takes place and it produces two structures, a diploid zygote and a primary endosperm nucleus. The zygote produces embryo, while as primary endosperm nucleus produces a nutritive tissue that is called endosperm, which provides nutrition to the growing embryo. With the growth of the embryo, the central part of the endosperm is consumed, and the remaining part of the endosperm in turn corrodes over the nucellus. As the embryo development reaches maturity, its further growth is suspended due to number first, development of inhibitors, number second, abscission of funiculus, and number third, change in the integuments. Now what happens to the integuments? The cells of the integuments enlarge, lose their protoplasm, develop thick and impermeable walls. The outer integument becomes hard and leathery and forms the outer seed coat called testa. The inner integument, if persists, forms the inner seed coat called tegmen. With this, ovule gets converted into a seed, and the tissues of the ovary wall also get stimulated with the development of the seeds. So they also grow and produce a fruit wall or pericarp. The growth of the fruit is generally very fast than the development of seed. So with the development of seed, fruit wall also gets developed and produce a fruit. Those fruits which are produced from ovary, they are called true fruits. While in some cases, thalamus and other flower they also proliferate and produce a fruit. Such fruits are called false fruits. For example, apple, which is produced from the thalamus. Two types of stimuli are involved in fruit formation. The first stimuli comes from pollination and the second stimuli comes from developing seeds. This is all about the development of seed and fruit. In another lecture, we will discuss about the biological significance of seed and fruit formation. This is all for today. Thank you. Assalamu alaikum.